Let's talk about cholesterol because this is one of the most misunderstood topics in human health. Cholesterol has been demonized for decades, yet it is absolutely essential for life. Every single cell membrane in your body requires cholesterol to function properly. Your brain is especially rich in cholesterol, and your body uses it to produce critical hormones like estrogen, testosterone, cortisol, and vitamin D. So right away, we need to clear up a major misconception. Cholesterol itself is not the enemy. The real issue is how cholesterol is transported in the body, and that's where the terms good cholesterol and bad cholesterol come from. These labels are simplifications, but they help us understand risk. Cholesterol does not travel freely in your bloodstream. It's a fat-like molecule, and blood is water-based, so cholesterol has to be packaged inside particles called lipoproteins. These lipoproteins act like delivery trucks, carrying cholesterol and other fats through the bloodstream to different tissues. The two most talked about lipoproteins are LDL and HDL. LDL stands for low-density lipoprotein, and HDL stands for high-density lipoprotein. LDL is commonly referred to as bad cholesterol, while HDL is called good cholesterol, but that language can be misleading if you don't understand what these particles actually do. LDL particles deliver cholesterol from the liver out to the cells that need it. That's a normal, necessary function. Your cells rely on LDL to maintain their membranes, repair damage, and support growth. The problem arises when there are too many LDL particles in the blood, or when those particles become damaged or oxidized. When that happens, LDL can penetrate the arterial wall and trigger an inflammatory response. Over time, this contributes to plaque formation, which is a key driver of atherosclerosis and cardiovascular disease. HDL, on the other hand, plays a very different role. HDL particles help remove excess cholesterol from tissues and from the walls of blood vessels and transport it back to the liver for recycling or elimination. This process is called reverse cholesterol transport, and it's one of the reasons HDL is considered protective. Higher levels of functional HDL are associated with a lower risk of cardiovascular disease, not because HDL is magically good, but because it supports cholesterol balance and vascular health. However, here's where things get more nuanced. It's not just about how much LDL or HDL you have. It's about particle number, particle size, and particle quality. Two people can have the same LDL cholesterol number on a standard blood test, but very different cardiovascular risk profiles. One person might have a small number of large LDL particles, while another has a high number of small, dense LDL particles. And the latter is far more strongly associated with arterial damage and plaque formation. Small, dense LDL particles are more likely to become oxidized, more likely to slip into the arterial wall, and more likely to provoke inflammation. This is one reason why metabolic health plays such a huge role in cholesterol-related risk, insulin resistance, chronic inflammation, and high blood sugar all increase the likelihood that LDL particles become small, dense, and dysfunctional. HDL has similar complexity. Simply raising HDL cholesterol numbers does not automatically reduce risk. What matters is how well HDL functions. HDL particles have antioxidant, anti-inflammatory, and endothelial protective properties when they are healthy, but in states of chronic inflammation, even HDL can become dysfunctional and lose many of its protective effects. Another critical point is that cholesterol levels do not exist in isolation. They interact with lifestyle, diet, sleep, exercise, stress, and genetics. Your liver produces the majority of the cholesterol in your body, and it adjusts production based on what your body needs. Diet does influence cholesterol, but not in the simplistic way we were once told. For most people, dietary cholesterol has a relatively small effect on blood cholesterol compared to factors like insulin, sensitivity, body composition, and inflammatory status. So when we talk about good and bad cholesterol, what we're really talking about is balance, context, and function. LDL is not inherently bad, and HDL is not automatically good. Problems arise when the system becomes dysregulated. In the next part, we're going to dive deeper into what actually causes LDL to become harmful, how inflammation and oxidation drive cardiovascular disease, and why focusing only on total cholesterol can be incredibly misleading. To really understand the difference between so-called good and bad cholesterol, we need to zoom out and talk about what actually damages arteries. Cardiovascular disease is not simply a problem of cholesterol buildup. It is, at its core, a disease of chronic inflammation combined with lipid dysregulation. Cholesterol becomes a problem only in the presence of an inflammatory environment. When LDL particles circulate in the bloodstream, most of them are doing exactly what they're supposed to do. They deliver cholesterol to cells and then return to the liver. But when the inner lining of the artery, known as the endothelium, becomes damaged, LDL particles can slip beneath that lining. This endothelial damage can be triggered by high blood sugar, smoking, high blood pressure, oxidative stress, lack of sleep, and chronic psychological stress. Once LDL enters the arterial wall, it is exposed to reactive oxygen species and becomes oxidized. Oxidized LDL is a major red flag for the immune system. The body recognizes it as a threat and sends immune cells, particularly macrophages, to clean it up. These macrophages engulf oxidized LDL and transform into foam cells. Over time, foam cells accumulate and form fatty streaks, which are the earliest visible signs of atherosclerosis. This process explains why LDL is often labeled bad. It's not because LDL exists, but because oxidized LDL in an inflamed artery drives plaque formation. The more LDL particles you have circulating, the greater the chance that some of them will become oxidized and trapped in the arterial wall. This is why LDL particle number is often a better predictor of risk than total LDL cholesterol. Now let's talk about HDL, because its role here is incredibly important. Healthy HDL particles can actually help counteract this process. HDL participates in reverse cholesterol transport, pulling excess cholesterol out of foam cells and the arterial wall and returning it to the liver. But HDL also has antioxidant enzymes that help prevent LDL from becoming oxidized in the first place. In this way, HDL acts as part of the body's cleanup and defense system. However, just like LDL, HDL can become dysfunctional. Chronic inflammation, obesity, insulin resistance, and smoking can alter HDL structure and reduce its protective abilities. In some cases, HDL may still show up as high on a blood test, but is no longer effectively removing cholesterol or protecting the endothelium. This is one of the reasons why simply chasing higher HDL numbers with medications has not consistently reduced cardiovascular events in clinical trials. Another layer to this story involves triglycerides. High triglycerides are often a sign of metabolic dysfunction and are strongly associated with small, dense LDL particles and low HDL. When triglycerides are elevated, it often indicates excess circulating energy, usually from refined carbohydrates, sugars, and insulin resistance. This metabolic environment makes LDL more atherogenic and HDL less protective. This brings us to a key point. Cholesterol problems are often downstream effects of poor metabolic health. Conditions like type 2 diabetes, metabolic syndrome, and non-alcoholic fatty liver disease dramatically alter lipoprotein behavior. In these states, the liver produces more VLDL particles, which eventually become LDL. And these LDL particles are more likely to be small, dense, and inflammatory. Physical activity plays a powerful role here. Regular exercise improves insulin sensitivity, lowers triglycerides, reduces LDL particle number, and improves HDL function. Exercise doesn't just change cholesterol numbers. It changes how lipoproteins behave in the body. Sleep quality and stress management also have measurable effects on lipid metabolism and inflammation. So when we label cholesterol as good or bad, we're oversimplifying a complex biological system. LDL becomes dangerous when combined with inflammation and oxidative stress. HDL is protective when it is functional and supported by a healthy metabolic environment. In the next part, we'll explore how diet influences cholesterol quality, which dietary patterns support healthy lipoproteins, and why some fats are far more beneficial than others for cardiovascular health. Now let's talk about diet, because this is where much of the confusion around cholesterol originates. For years, dietary fat and dietary cholesterol were blamed as the primary drivers of heart disease. But when you look closely at the biology and the long-term data, the picture is far more nuanced. What you eat doesn't just affect how much cholesterol you have, it affects the type of lipoproteins your body produces and how they function. First, dietary cholesterol itself has a relatively small impact on blood cholesterol levels. For most people, your liver tightly regulates cholesterol production. When you eat more cholesterol, your liver usually makes less. When you eat less, your liver makes more. This feedback system explains why foods like eggs have minimal effects on LDL cholesterol in the majority of metabolically healthy individuals. What matters far more is the overall dietary pattern, especially how it influences inflammation, insulin sensitivity, and oxidative stress. Diets high in refined carbohydrates and added sugars tend to increase triglycerides, promote insulin resistance, and shift LDL particles toward the small, dense, more atherogenic type. This metabolic environment also impairs HDL function, reducing its ability to remove cholesterol from the arterial wall. In contrast, diets rich in whole, unprocessed foods tend to improve lipid profiles in meaningful ways. Fiber plays a particularly important role. Soluble fiber, found in foods like legumes, oats, berries, and vegetables, binds bile acids in the gut and helps remove cholesterol from the body. Fiber also feeds beneficial gut bacteria, which produce short-chain fatty acids that improve insulin sensitivity and reduce inflammation. The type of fat you consume also matters. Monounsaturated fats, such as those found in olive oil, avocados, and nuts, are consistently associated with improved LDL particle size and better HDL function. These fats reduce oxidative stress and support endothelial health. Omega-3 fatty acids, particularly EPA and DHA found in fatty fish, are especially powerful. They lower triglycerides, reduce inflammation, and may stabilize plaques, making them less likely to rupture. On the other hand, industrially produced trans fats are clearly harmful. They increase LDL, decrease HDL, promote inflammation, and impair endothelial function. These fats directly worsen every aspect of lipid metabolism and cardiovascular risk. Highly processed seed oils that are repeatedly heated can also contribute to oxidative stress when consumed in excess, particularly in the context of a nutrient-poor diet. Saturated fat is more complex and often misunderstood. Saturated fat can raise LDL cholesterol in some individuals, but the effect varies significantly based on genetics, metabolic health, and the food source. Saturated fat from whole foods like dairy or unprocessed meats behaves very differently in the body compared to saturated fat consumed alongside refined carbohydrates and sugars in metabolically healthy individuals. Saturated fat often increases LDL particle size rather than particle number, which may be less harmful. Another critical dietary factor is micronutrient density. Nutrients like magnesium, potassium, vitamin K2, and antioxidants from colorful plant foods support vascular health and reduce the likelihood that LDL becomes oxidized. Polyphenols found in foods like berries, green tea, dark chocolate, and extra virgin olive oil have been shown to improve endothelial function and reduce oxidative stress. Timing and energy balance also matter. Chronic overeating, regardless of macronutrient composition, leads to excess triglycerides and lipid dysfunction. Intermittent fasting or time-restricted eating, when done appropriately, can improve insulin sensitivity and lipid profiles in some individuals, though it's not a universal solution. So diet doesn't simply raise or lower cholesterol, it shapes the entire metabolic context in which cholesterol operates. A diet that reduces inflammation, supports insulin sensitivity, and provides antioxidant protection allows LDL and HDL to do their jobs without causing harm. In the next part, we'll examine how lifestyle factors beyond diet, including exercise, sleep, and stress, dramatically influence cholesterol behavior and cardiovascular risk. Beyond diet, lifestyle factors play an enormous role in determining whether cholesterol behaves in a protective or harmful way. Exercise, sleep, stress management, and even sunlight exposure all influence lipid metabolism, inflammation, and vascular health. These factors often have effects that are just as powerful, if not more powerful, than dietary changes alone. Let's start with physical activity. Regular exercise is one of the most effective ways to improve cholesterol quality. Aerobic exercise, resistance training, and even regular walking increase insulin sensitivity, lower triglycerides, and reduce LDL particle number. Exercise also shifts LDL particles toward a larger, less atherogenic form and improves HDL functionality. Importantly, exercise enhances the antioxidant capacity of HDL, making it more effective at preventing LDL oxidation. Exercise also directly improves endothelial function. The increased blood flow during movement creates shear stress along the artery walls, which stimulates the production of nitric oxide. Nitric oxide helps blood vessels relax, reduces inflammation, and prevents LDL from sticking to the arterial wall. This is one of the reasons physically active individuals have lower cardiovascular risk, even when their cholesterol numbers don't look perfect on paper. Sleep is another major and often overlooked factor. Chronic sleep deprivation disrupts glucose metabolism, increases cortisol, and raises inflammatory markers. Even short-term sleep restriction has been shown to increase insulin resistance and shift lipid profiles in an unfavorable direction. Poor sleep can increase LDL and triglycerides while impairing HDL function. High-quality sleep, on the other hand, supports hormonal balance and allows the liver to regulate cholesterol production more effectively. Stress plays a similar role. Chronic psychological stress elevates cortisol and adrenaline, which increase blood sugar and promote inflammation. And over time, this stress-driven metabolic state increases LDL particle number and triglycerides and reduces HDL functionality. Stress also directly damages the endothelium, creating the conditions that allow LDL to penetrate the arterial wall. Practices like mindfulness, breathing exercises, and regular physical activity can measurably improve lipid markers by reducing stress-induced inflammation. Sunlight and vitamin D are also part of this conversation. Vitamin D is synthesized from cholesterol in the skin in response to UVB exposure. Low vitamin D levels are associated with higher cardiovascular risk, increased inflammation, and poorer lipid profiles. While vitamin D is not a cure-all, adequate levels support immune regulation and vascular health, which indirectly influence how cholesterol behaves. Another important factor is smoking and environmental toxin exposure. Smoking dramatically increases oxidative stress and directly oxidizes LDL particles. It also lowers HDL levels and impairs HDL's protective functions. Even exposure to air pollution has been linked to increased inflammation and higher cardiovascular risk through similar mechanisms. Reducing toxin exposure wherever possible is a meaningful way to protect vascular health. Alcohol deserves a brief mention. Moderate alcohol consumption has been associated with higher HDL levels, but this does not mean alcohol is necessary or recommended for heart health. Excess alcohol raises triglycerides, damages the liver, and increases inflammation. Any potential benefit is quickly outweighed by harm when intake is excessive or poorly tolerated. Genetics also influence how lifestyle factors interact with cholesterol. Some people are more sensitive to saturated fat, others to carbohydrates, and others have genetic variations that affect lipoprotein production and clearance. However, even with genetic risk, lifestyle interventions can significantly reduce cardiovascular events by improving inflammation and metabolic health. All of this leads to a critical takeaway. Cholesterol numbers do not define destiny. They reflect the state of your overall physiology. Improving sleep, moving regularly, managing stress, and reducing inflammatory exposures can dramatically change how LDL and HDL function in the body. In the final part, we'll bring everything together and talk about how to interpret cholesterol tests intelligently and what truly matters when assessing cardiovascular risk. Now let's bring everything together and talk about how to actually interpret cholesterol in a meaningful way. One of the biggest mistakes people make is focusing on a single number, like total cholesterol or LDL cholesterol, without considering the broader context. Cholesterol is not a standalone villain or hero. It's a marker within a complex biological system. A standard lipid panel typically includes total cholesterol, LDL cholesterol, HDL cholesterol, and triglycerides. While this provides some useful information, it does not tell the whole story. Total cholesterol is simply a sum of different lipoprotein-associated cholesterol and has very limited predictive value on its own. LDL cholesterol gives an estimate of how much cholesterol is carried inside LDL particles, but it does not tell you how many LDL particles there are or how they behave. This is where more advanced markers can be helpful. LDL particle number, often measured as ApoB, is a stronger predictor of cardiovascular risk than LDL cholesterol alone. Each atherogenic particle carries one ApoB molecule, so ApoB gives a direct estimate of how many potentially harmful particles are circulating in the bloodstream. A higher particle number increases the likelihood that some of those particles will penetrate the arterial wall and become oxidized. Triglyceride levels also provide important insight. Elevated triglycerides often indicate insulin resistance and metabolic dysfunction, which strongly influence LDL particle size and HDL function. Low triglycerides combined with higher HDL levels generally reflect better metabolic health. The triglyceride to HDL ratio is a simple metric that can offer clues about insulin sensitivity and cardiovascular risk. It's also important to understand that lowering LDL cholesterol does not always address the root cause of disease. If inflammation, poor sleep, chronic stress, and insulin resistance remain unaddressed, cardiovascular risk may persist even with improved cholesterol numbers. This is why lifestyle interventions consistently reduce cardiovascular events, sometimes more effectively than focusing on cholesterol alone. That said, medications like statins can be life-saving for certain high-risk individuals. Statins reduce LDL particle number and also have anti-inflammatory effects. For people with established cardiovascular disease or very high risk, these medications can significantly reduce the likelihood of heart attacks and strokes. The key is understanding that medications are tools, not substitutes for addressing underlying metabolic health. The most protective strategy is one that improves cholesterol function, not just cholesterol levels. This includes eating a nutrient-dense, anti-inflammatory diet, maintaining regular physical activity, prioritizing sleep, managing stress, and avoiding smoking. These interventions improve endothelial health, reduce LDL oxidation, and enhance HDL's ability to remove excess cholesterol. Ultimately, the difference between good and bad cholesterol is not a simple label. LDL becomes harmful in the presence of inflammation and oxidative stress. HDL is protective when it is functional and supported by a healthy lifestyle. Cholesterol reflects how well your body is regulating energy, inflammation, and repair. When you shift the focus from fear to understanding, cholesterol becomes a powerful window into your overall health. Instead of asking how low your cholesterol can go, the better question is how resilient your metabolism and vascular system are. When those systems are supported, cholesterol works the way it was always meant to, as a vital molecule that supports life, not one that shortens it.